Welcome to the SMEC Veterinary Diagnostic Lab Video 3. This video will describe what happens to your samples as they are processed through the diagnostic lab. Delivery truck arrival. Each morning around 9 o'clock, the delivery trucks arrive with packages shipped from practices and farms the day before. The diagnostic lab receives between 60 to 100 boxes per day, and most of these boxes contain more than one case. It is important when shipping that you use a service that will get your packages to the diagnostic lab first thing in the morning. Several tests have an 11:30 a.m. cutoff for same-day service, so packages arriving later in the day may delay your results. First off, the diagnostic lab sorts through the packages to determine whether they go to the serology or pathology mailroom. Serology mailroom. In the serology mailroom, the boxes are opened and they make sure the samples are numbered in plastic snap cap tubes, such as a falcon tube. If they are improperly submitted or in glass tubes, they must be centrifuged again and poured off under the fume hood. The diagnostic lab policy is to not allow glass tubes to leave the mailroom because of the increased cross-contamination of the rubber stopper tops. Each sample must be placed in a plastic snap cap tube. Obviously, this extra processing slows down the submission process and doesn't help you get faster results. Each serology case receives an accession number which follows the case through the lab. We will be discussing this numbering system in a few moments. Once the serum is properly prepared, it is sorted and placed on this cart to be sent to the appropriate department, such as serology or molecular and viral diagnostics. All other cases are sent to the pathology mailroom. This includes samples of tissue, whole blood, feces, urine, ear notches, and oral fluids. Each box is unpacked and checked to make sure the proper samples and paperwork are included. If there is mismatched or no paperwork, leaking or improperly packaged specimens, or animal IDs not provided, this case is set aside to be dealt with later. Usually a phone call to the practitioner needs to be made to fill the gaps in knowledge of missing information. Once again, you can see how properly submitting your samples accelerates the process and helps you get timely results. This is a copy of a submission form, much like you would fill out at your clinic. Once unpacked, each case receives an accession number and is entered into the computer system. A sticker is placed on this form and all other components of the case, such as tubes or tissue trays. This allows the case coordinators and staff to see where the samples are at within the testing process at any given time. Part of this accession number is the case number you will see when checking results or asked to have available if you call the diagnostic lab with questions. Each submission form is then scanned into the system. The ISU Veterinary Diagnostic Lab uses a computer program called LIMS, or Laboratory Information Management System, which allows for tracking of each case in a paperless manner. After this time of triage, the staff in the pathology mailroom routes the specimens to the proper area. If the case requires a pathologist review, it is provided to the technicians and the pathologist on the necropsy floor. You can see the window that allows the pass-through of cases to the floor. If a test is clearly requested on a specimen, such as PCR on oral fluids, the case is then routed through the laboratory to its specific department. Once the cases are sorted by the staff, they are transferred to the diagnostic lab floor. The submissions are reviewed and processed by the technical staff. The diagnostician on duty reads the case history, examines the submitted tissues, and makes the best decision on which diagnostic test to run. These test requests are entered into the computer on the floor, continuing the paperless process. They are then sent to the appropriate departments. Besides gross pathology on the necropsy floor, most submitted tissues go through the histopathology department. When you send formalin-fixed tissues to the diagnostic lab, they are processed through here. The formalin containers are slid through this window to the trimming room. They are trimmed into pieces about 3 millimeters in size and placed in these small cassettes, which are about 1 inch square. It is important to remember, as we stressed in video 2, that a formalin-fixed tissue sample needs to be no thicker than 1 centimeter for proper fixation purposes and because it will be sliced into even thinner portions. These cassettes are then placed overnight in this processor that dehydrates the tissues and infiltrates them with paraffin. Once the tissues have been embedded in paraffin, they are in block form and ready to be processed the next day. This allows them to be sliced into very thin sections on a special cutting machine called a microtome. Each section is about 4 microns thick. Remember that a red blood cell is 7 microns in diameter, so these slide sections are truly getting to one cell layer of thickness. The section is then placed on a slide and into a water bath to remove the paraffin and rehydrate the slide for staining. Almost all slides receive a hematoxylin and eosin stain. When the stains are done, a cover slip is placed on the slide and it is delivered to the pathologist investigating the case. Generally, when a pathologist processes a case on the necropsy floor, he or she has the histopathology slides by 10 a.m. the next day. If immunohistochemistries or special stains are requested, they are added and those slides are ready just a few hours later. Bacteriology. Most cases have a component to filter through the bacteriology department. Tissues are taken as quickly as possible to the lab for culture on various types of media. They are then examined for growth at 24 hours. Further culture and testing, along with antimicrobial MICs, may be performed after this point. Other areas where your cases may be submitted to are molecular and viral diagnostics, which includes PCRs, virus isolation, fluorescent antibody and other tests, toxicology, or FAST, the Pharmacology Analytical Support Team, which runs racing chemistry and antibiotic residue testing. The ISU Veterinary Diagnostic Lab has an extensive quality assurance program to ensure testing methods and your results are held to the highest standards. It is essential to maintain proper lab function and providing you with accurate results. The lab is accredited by the American Association of Veterinary Laboratory Diagnosticians, or AAVLD. This accreditation involves an in-depth, week-long process of on-site surveillance and documentation once every 5 years. In addition, the ISU Veterinary Diagnostic Lab performs its own internal audits twice a year, using many of the same standards set by the AAVLD for accreditation. Each department has quality assurance standards that are always ongoing. These are the areas mentioned before: serology, necropsy, which covers histopathology, molecular diagnostics, microbiology, clerical, and information technology. Standards include compliance to procedures and policies, proper documentation, and proficiency training in new procedures, equipment, and disease information. There is also an entire system in place to monitor complaints, non-compliant issues, and non-conformancies, or any testing parameter that does not meet with standard operating procedure. For example, a computer program keeps track of each test result, customer feedback, test reporting, client submissions, and equipment. You can see here this illustration of graphs charting the diagnostic controls for PERS testing. This allows the lab to monitor function and take any corrective or preventative actions. Although the quality assurance program may not be the first area that comes to mind when you use the diagnostic lab, it is essential to proper lab function. Virtually every testing parameter and procedure is monitored to provide you with accurate and high-quality results. This concludes Video 3, demonstrating the processing of cases through the Iowa State University Veterinary Diagnostic Lab. For more information on the Swine Medicine Education Center and the Veterinary Diagnostic Lab, please feel free to contact us.